The overall goal of this procedure is to test and confirm herbicide resistance in a wide range of weed species and herbicides, as well as to infer the herbicide resistance levels. This is accomplished by first breaking the dormancy of seeds collected from plants that had survived an herbicide treatment. To obtain simultaneous germination and seedling emergence, a period of seed vernalization ranging from a few days to more than a week is required. The second step is to pour an agar medium with potassium nitrate into plastic dishes. The vernalized seeds are then placed on the solidified agar, and the dishes are transferred into a germination cabinet with the optimum conditions for each weed species. Next, 15 to 20 seedlings are transplanted into plastic trays filled with a standard potting mix. A barcode is assigned to each tray, including all information for its unique identification, and the trays are placed in a heated greenhouse until seedlings reach the 2 to 3 leaf stage. The final step is the herbicide treatment using a precision bench sprayer. All herbicides are applied as commercial formulations with recommended surfactants at 2 doses, recommended field dose, and 3 times that. 3 to 4 weeks after the treatment, the final assessment is done. A barcode reader is used to record the number of survived plants and the visual estimated biomass per experimental unit. Data are then downloaded into a computer and analyzed to determine the herbicide resistance status.
The main advantage of this method over other in vivo or in vitro diagnostic screening tests is related to its robustness and flexibility. In other words, the method is reliable and can be easily adapted to a wide range of species through minor adaptations.
This method helps answer